Okay, class, we're going to start off where we left off last time. We were looking basically at cell-cell adhesion, and we left off with the synaptic junctions. And so now we're going to look at sort of the tight junctions, and then we'll get into the basal lamina and the extracellular matrix, and also some of the gap junctions. But in general, in sort of a epithelial layer, at the very top, you have these tight junctions, and they prevent basically small molecules and stuff from passing between. And we saw last time with sort of the transcellular transport that, things that requires then things sort of go through the cell because these tight junctions are going to sort of prevent like glucose from going down its concentration gradient. So you'll have glucose and then the sodium-driven glucose symporter, which will bring in glucose against its concentration gradient, and then the glucose can actually just sort of fall and pass through the passive glucose carrier. And so it's ultimately these tight junctions that require that sort of necessitate that you have this sort of transcellular transport. So here's a nice little schematic of the tight junction, and then you can see these sort of EMs where they're sort of connected, and here's another one. And these really sort of affect the permeability between the cells so that, the tracer molecules like this one out here cannot pass through. And so this sort of makes it so the cells are permeable to ions, but less so for food sources. And then in the case with sort of where your kidneys are, it's less so for the kidneys, the permeability to ions, right? And that allows us to filter out all of the salts and stuff out of our bloodstream that we don't want. So here's just a freeze fracture EM of the tight junctions. You can see how they're sort of connected here, and here's just another EM where you have sort of all of the contacts between these tight junctions. The way they're set up is between these molecules called claudin and occludin. And so here's just a schematic of claudin and occludin. And what they do is almost zipper the cells together, or that's sort of how I think of it. They're sort of almost sewn together here so that they create this mesh that doesn't allow stuff to pass through them. There's over 24 claudins in humans, but they're sort of set up like this. And then they sort of zip the two different cells together. Here's just another EM of the tight junctions. And once again, this is really sort of to emphasize the hierarchy that I was explaining before, where you have your tight junctions on top, and then your adherent junctions. These are going to have actin attachments, and then your desmosomal junctions, which are going to have your sort of intermediate filaments or keratin, so sort of attached to them. So once again, tight junctions on top, adherens next, and the desmosomes under them. The tight junctions in mollusks and invertebrates actually forms a septate, and this is just a really cool image. Nothing that you guys really need to know here, but it's just kind of a pretty image here. And it just sort of goes to show their repetitive nature. These also help establish polarity, and there's a small G protein called RAC that also helps this. Okay, so normally you'll have apical to basal polarity with sort of the actin on top, right, and then the attachments to the laminin on the outside. If you block RAC function, the polarity can actually change, and then if you sort of overexpress laminin on the outside here, and even if RAC is sort of function is blocked, it'll still form in the right polarized manner, right? So it's a combination not only of the hierarchy that's there, but also what's sort of can the cell bind to, which sort of affects it. So there'll be the tight junctions, your adherent junctions, your desmosomes, your gap junctions, and then the hemidesmosomes and the attachments to the lamin, the extracellular matrix or the laminin. Here is just the cell polarity again. So this is if you are lacking this LKB1, but if it's active, you'll start to sort of have these actin-filled microvilli on the outside. So once again, there's a lot of things that are sort of involved in establishing the polarity of the cell. And here you can sort of see it again. You're going to have your tight junctions on top, and then near that will be the RAC, the small G protein, then your adherens junctions, and then your desmosomal junctions, and then ultimately down on the bottom you're going to have your hemidesmosomes attached to the basal lamina. And it's pretty much the same in Drosophila, but you don't need to necessarily know the Drosophila ones. But and here's a good example of planar cell polarity. And so this is basically epidermal cells in the flywing. And normally they're all sort of polarized, so the sensory hairs are sort of facing in one direction. But if you have flamingo, a mutant called flamingo, they sort of go all over the place, and you can see that the sensory hairs are all pointed differently. So it turns out flamingo is actually one of the cadherin family members. We saw it in the schematic. If you guys want to go back to part 1, you can sort of find it. But once again, if there's a mutation in it, that's going to affect the polarity. And once again, flamingo, frizzled, and disheveled are all sort of part of Wnt signaling. So you can get this sort of lack of polarity in those as well. Okay, now on to gap junctions. So the gap junctions just make small little pores, small molecules with a low molecular weight, 100 to 1,000, can freely pass from cell to cell. However, when they get larger, like the size of small proteins and even medium-sized or even small medium-sized proteins can't pass. Okay, but they create a small passageway for gases and other small molecules to pass through. The gap junctions are made by these things called connexons. Here they are. There's a total of 6 of them. They sort of form this hexameric complex, and then they sort of come from cell to cell. So if you look right here, you see 2 connexons sort of form this pore that small molecules can fit through. They can come in all shapes and sizes. You have the different connexons. They can be homomeric or heteromeric, and homotypic and heterotypic, and so they just form these small channels. You guys don't really need to worry too much about these. Just know that there's these sort of channel junctions that occur in cells. Here they are, again, the gap junctions. Here's a large patch of them. Here's a much smaller patch of them, and just small molecules can pass freely from cell to cell through these. And they get turned over depending on how much the cell wants to signal. So here's a cross-section of it, and then after 4 hours, you can start to see after a fair amount of incubation, it gets shorter, and this is sort of a top-down view of it. You can see they're different sizes. And these actually affect the permeability of the cell. So this is just a tracer going through some nerve cells, and then when you add some dopamine in here, it'll actually reduce the flow, and it's all sort of confined to just one neuron here, and it doesn't pass to other neurons. Okay, so once again, the permeability of the gap junctions can actually be controlled. Okay, so then onto the basal lamina and all the connective tissue. Okay, so here's just some cells, some muscle cells, and the muscle cell plasma membrane, and these are all sort of connected to connective tissue and the basal lamina. and it's sometimes referred to as the basement membrane, but these have sort of extracellular proteins, and they're connected via sort of the integrins and things like that. So once again, here's your epithelium, and then you have your basal lamina, the basement membrane, and then under that is the connective tissue. In the kidney, it sort of goes between the blood and the endothelial cells, and then you'll have your epithelial cells and your basal lamina here. And so this is what sort of separates the blood from the urine, and sort of small, these can be attached so certain ions can pass and other things can't. So, and the major protein of the basal lamina is this protein called laminin, which we'll see in a second here. Here's just a nice sort of EM, and you can see the basal lamina is this sheet, and then you have your epithelial cells, and then in the extracellular matrix you have a lot of the collagen and things like that. So this is just an EM of the basal lamina. Here's all the extracellular matrix macromolecules. You should sort of be familiar with some of these. You know, definitely know laminin and type 4 collagen. We've sort of covered these, but there's also fibronectin and some other ones. Here's the laminin, the sort of the structure of it, and it forms this triple helix here with the coiled domain, and it's 3 chains: the alpha, the beta, and the gamma. And these sort of self-assemble and can attach to integrins or other things in the sort of extracellular matrix, like type 4 collagen and things like that. Here's just an EM of it. Once again, you can see the coiled coils and sticking out. And if you sort of look at it in a schematic, this is what the basal lamina is going to sort of look like, where you have laminin and perlecan and some of the other things, some collagen wrapped through here. The collagen's in red, and then the integrins will actually attach to the laminin and other things, and that helps hold the cell in place. So so you can see the plasma membrane. So you're sort of looking down on it now. And so all the information in the basal lamina helps sort of cellular connections. So if you have a muscle cell and a nerve cell attaching, and here you have your sort of synaptic junction here, if you sort of kill the muscle cell and kill the nerve cell, sort of the residual basal lamina with the junction that's there will sort of maintain. And then if you put a nerve cell back in, it'll know, the nerve cell will know where it needs to attach. And likewise, if you sort of regenerate the muscle will be right there. And so it sort of helps to organize cell-cell attachments as well. Okay. Now, the integrins actually attach to the extracellular matrix. So here's, and we saw that before, where you can see these integrins sort of connecting to the lamina. But here's the integrins again. When they're active, they're sort of bound. In this case, there's an alpha and a beta subunit. And then they're sort of connected to actin filaments via talin and vinculin here. And so that helps them grab onto things and hold them in place. Okay, so this is basically an actin anchoring junction here with the integrins. You can also have them with the hemidesmosomes, where you have, you know, your intermediate filaments, your keratin, plectin, and dystinin here, and then you've got your integrins attaching to the laminin, and then down to the collagen. And that's just sort of shown in this schematic over here. So you've got the basolateral surface of the cell attaching to the basal lamina. The integrins are regulated, and we sort of saw this with the rolling leukocyte. When it starts to crawl, it'll get a signal, and then it becomes sort of get a much stronger attachment. And so you can have the signaling coming from two locations. You can have outside in activation or inside out activation, and it depends on just where the signal's coming from. But then once you get the signal, you get sort of strong ligand binding on one end and then strong talon binding, which would then be connected to the actin to sort of hold the cell in place. And that's for a dynamic cell that's sort of moving along or rolling. And so here's sort of the inside out activation. You can have it occur through a receptor tyrosine kinase or a G protein coupled receptor. They'll eventually signal to talon, which then talon becomes activated and sort of serves as a scaffold between the active integrins and the actin filament. So here's just some of the different types of integrins. No need to know this, but just sort of be aware that, you know, they can attach to laminin or fibronectin, or, you know, we saw in the last lecture the sort of the IG superfamily like ICAM and things like that. So there's just a numerous different types of integrins that are expressed. And they're also involved in antigen presentation and immunological synapses. Okay, so up here you'll have your T cell, and then you'll have your antigen presenting cell like a macrophage, and if they're presenting an antigen with the MHC class 2 or class 1 molecules, they're presented to the T cell. If there's a good match between the T cell receptor, there's actually signaling to the integrins and ICAM, and they'll form a much tighter connection and hold it there so that then you can start to have an immune response. All right, there's a sort of a signaling, and then you can get things like T cell expansion and other things. They're also really important in cell spreading and things like that. And we saw that with the lamellipodia formation. But if you just put like a little defined patch of fibronectin on a slide and you put a cell there, the cell's going to die of apoptosis. It really wants to grow and spread. And so if you just put little patches all over the place and then you were to put a cell on it, the integrins can find that fibronectin and actually sort of use it and stretch, and that's what's called anchorage dependent growth. And if you've ever grown tissue culture cells under, up, you'll oftentimes see them roll up and ball up and float away, whereas the nice healthy cells are attached to the plate and sort of growing and spreading outward. And sort of that's regulated by this protein called focal adhesion kinase, and you can see the actin filaments here, and then the phosphotyrosine is stained in orange here. All right, so just a kinase here, it's adding phosphates, and this helps that sort of growth where the attachment sites are going to actually have integrins, and then they'll be bound to fibronectin or they'll laminin or something else on the surface. And so the focal adhesion kinase is actually required for spreading. So here's a normal fibroblast, and it's spreading. And if you stain the cell for integrin and vinculin, you can see if you have a FAC deficient fibroblast, it'll just roll up, and even though the integrins are there, they can't attach to anything. Okay, so here's just a big overview of some of the cell adhesion molecules, and for the interests of what I really want you guys to know, make sure you know the cadherins and how they function. I went over them in great detail, and know the difference between the adherins or the desmosomes. Don't necessarily need to know all the players, but just know that the different junctions are there, and that's some of the big ones. Some of the IG family members, you don't necessarily have to know exactly what's what, but just how they all connect and everything like that. And then how they're attached to, how the cell attaches to the extracellular matrix through the integrins and things like that. Okay? So that's it for today. So next week we're going to do mitochondrial transport and electron transfer, a real sort of favorite topic of mine, unlike today, which I kind of find cell-cell adhesion and cell attachment a little on the boring side. There's really just not much to know there. You just need to sort of understand the cadherins and the integrins and things like that. And it's got a big place in sort of cancer and metastasis, as I mentioned in the beginning, because once again, cancer cells figure out a way to get around that and escape and sort of relocalize to different portions of the body. So for your reading for next time, chapter 12, pages 713 to 714 in the 5th edition, and chapters 12 and 14 in the 6th edition.